Melting permafrost can release radioactive gas. As the climate warms, huge areas that were previously covered with ice are beginning to thaw. Melting permafrost, as scientists have determined, can release radon, a colorless and odorless radioactive gas that can cause cancer. According to a new study published in Earth Science Reviews, the melting of permafrost may pose an increased health risk to people living in subarctic communities due to the release of radon. It is a gas produced by the decay of radium. It is odorless and colorless and may cause cancer. The melting of permafrost caused by climate change covers increasingly larger areas. This process can release microorganisms that have been dormant for thousands of years, including pathogens that can infect modern organisms. But permafrost also serves another function. It acts as a protective barrier against gas storage facilities located underground. The most famous of them is methane, a powerful greenhouse gas, the release of which further accelerates climate change. But there is also another gas lurking underground: radon. Progressing warming may create conditions that allow radon to reach the surface. This is a potential problem because radon can cause cancer. This gas is known to sometimes accumulate in homes, especially basements, thereby increasing the long-term risk of developing cancer. Radon is known to be the second leading cause of lung cancer in the US after smoking. According to the local Environmental Protection Agency, it causes 21,000 deaths per year. Cigarette smoking increases the risk of lung cancer acquired from exposure to radon by approximately 26 times. Additionally, smoking is 4.4 times more common in Arctic communities. Therefore, the release of radon may pose a health risk. In the new study, scientists collected and analyzed previous work on radon release in regions where permafrost occurs. They covered the area of Alaska and even northeastern China. This review suggests that permafrost degradation may allow radon to migrate into the basements of homes and workplaces, but, as the study's authors added, data on radon migration in permafrost areas is grossly insufficient. The permafrost does not melt evenly. Seismic activity, which is common in Alaska, can cause cracks and fissures to form in the ground through which radon can escape. With such uneven melting, you can't really tell where the radon will ultimately break through, said Art Nash of the University of Alaska Fairbanks. There are also questions about how radon interacts with other gases trapped underground. How will these gases compete for the limited routes that will be opened in the permafrost? Nash added, if the permafrost was stable, there would be no cause for concern. However, it is now widely accepted that climate change is leading to significant thawing of permafrost. And permafrost loss in the Arctic region is expected to be approximately 42% by 2050. At least this is what results from research from 2 years ago. Microplastics detected in every placenta tested. After analyzing 62 placenta samples, scientists said they found microplastic particles in every sample tested. This work shows how common contamination is with tiny pieces of plastic. Microplastic pollution is taking on alarming proportions. These tiny plastic particles have been found almost everywhere on the planet. It is present in the air, water and soil. The ubiquitous contact with plastics causes pieces of them to enter our bodies. In research from 2022, scientists showed that we consume up to 5 grams of microplastics a week. Therefore, it is not surprising that pieces of plastic are found in the blood, lungs and hearts. Generally, microplastics can accumulate in all organs, including the brain, and can cause changes in behavior. In a new study, scientists found pieces of plastic in every human placenta they examined. And although these microplastics were first discovered in placentas in 2020, the current study aimed to estimate how much and what types of plastic could be found in them. The description and results of the study were published in the journal, Toxicological Sciences. Since the early 1950s, plastic consumption around the world has increased exponentially, translating into thousands of tons of waste ending up in landfills and oceans each year. In landfills, but more so in the oceans. These plastics begin to crumble into smaller and smaller pieces under the influence of ultraviolet radiation in sunlight and the constant friction caused by waves. This is how microplastics are created. All this aerosolizes over time. Microplastics enter our bodies not only through ingestion, but also through inhalation. This affects not only us humans, but all animals and plants, said Marcus Garcia, co-author of the publication. Behind the new research are scientists from the University of New Mexico, UNM, who collaborated with colleagues from Baylor College of Medicine and Oklahoma State University. They began their work by collecting placenta samples and breaking down the fat and proteins they contained in a process known as saponification. They then spun each sample in a centrifuge, which left small pieces of plastic at the bottom of the tube. The results of these activities resulted in the detection of microplastics in all 62 placenta samples tested at concentrations ranging from 6.5 to 790 micrograms per gram of tissue. While these numbers may seem small, a microgram is a millionth of a gram. There is no real way to know what health effects it may have. According to toxicologists, the dose makes the poison, and the amount of microplastics appears to be constantly increasing. Traditionally, plastics are assumed to be biologically inert. But some microplastics are so small that they are measured in nanometers, 1 billionth of a meter, and are able to penetrate cell membranes. In the next step, researchers placed the plastic granulate in a metal cup and heated it to 600 degrees Celsius. And then recorded the gas emissions resulting from the combustion of various types of plastics at specific temperatures. The gas emission goes to the mass spectrometer and gives a specific fingerprint, said Dr. Matthew Campen of UNM, who led the study. It's a really cool method, he added. The researchers found that the most common polymer in placental tissue was polyethylene, which is used to make plastic bags and bottles. This type of plastic accounted for 54%. Total plastic particles found in bearings. polyvinyl chloride, better known as PVC, and nylon made up about 10% total, and the remainder consisted of 9 other types of plastics. According to Kampen, the microplastics we see in the environment are probably 40 to 50 years old. The researcher also said that rising concentrations of microplastics in human tissues may explain the puzzling increase in the incidence of certain types of health problems, such as inflammatory bowel disease and colorectal cancer, in people under 50. According to him, this may also explain fertility problems. The high concentration of microplastics in the placenta is particularly concerning, Kampen said. He explained that the placenta begins to form about a month after the beginning of pregnancy, i.e., it develops for a maximum of 8 months, and still during this time large amounts of plastic particles accumulate in it. What then about other organs that are with us for a much longer time? The problem of plastic pollution has been known for a long time. However, the production of plastics has not decreased at all, but is still increasing. Unknown seamounts and dozens of new species have been discovered at the bottom of the Pacific Ocean. Scientists from the Schmidt Ocean Institute, while exploring the sea depths off the coast of Chile, came across previously unknown underwater mountains, the highest of which rises to a height of 3,530 meters above the seabed. Around them, they discovered diverse ecosystems and dozens of never-before-seen species. Oceans cover about 70% Earth's surface, but only a small fraction of the area they cover has been explored. Scientists from the Schmidt Ocean Institute are trying to deepen our knowledge of the ocean depths. In a recent expedition off the coast of Chile led by Dr. Javier Solanes from the Universidad Católica del Norte, they encountered dozens of new species living around underwater mountains. Researchers from the Schmidt Ocean Institute have already made some amazing discoveries. Last year, they found an incredibly rich ecosystem full of strange species of invertebrates, snails and deep-living octopuses under hydrothermal vents at the bottom of the Pacific Ocean. The new research expedition also brought revelations. Experts say there are up to 100 species of deep-sea corals, sponges, sea urchins, amphipods, lobsters and others that are likely new to science. The research expedition focused on the depths off the coast of Chile. During the expedition, scientists used an underwater robot that can go to a depth of 4,500 meters to check what is happening around the underwater mountains lying at the bottom of the Pacific Ocean near the island of Salas y Gomez. These mountains belong to a chain stretching 2,900 kilometers from the coast of Chile to Rapa Nui, or Easter Island. During their work, scientists prepared a map of the local seabed, covering a total area of over 52,000 square kilometers. Mapping the seabed revealed 4 previously unknown underwater mountains, the highest of which is 3,530 meters above the seabed. The research team named it Salito, but this is an unofficial name. Seamounts are usually extinct volcanoes with steep slopes. Their peaks are deep underwater. It is estimated that the seabed is dotted with such structures, but only a small part of it has been known so far. Underwater mountains are habitats for a variety of life forms. They provide animals with space to live, shelter and food. Scientists found distinct ecosystems on each mountain they studied, many of which are extremely sensitive and possibly endangered, such as deep-sea coral reefs and sea sponge gardens. It is a fascinating world with a variety of shapes and colors, inhabited by strange-looking creatures, including the mentioned multicolored sponges, spiral corals, strangely elongated sea urchins and lobsters with peculiar beads. Some of them are probably unknown to science. We significantly exceeded our expectations for this expedition. You always expect to find new species in these remote and underexplored areas, but the amount we found, especially for certain groups like sponges, is staggering, Solanes said. Scientists analyzed the collected data. According to them, many of the specimens found are new, but full identification of the contents of the samples may take many years. The team carefully analyzes the physiology and genetics of the specimens to confirm whether they are indeed new to science. On February 24, a new Schmidt Ocean Institute expedition started. Scientists intend to return to the vicinity of Salas y Gomez Island and explore the depths of the southeastern Pacific throughout 2024. High-resolution photos and other materials from the research expedition can be seen on the Schmidt Ocean Institute website. Why don't people have tails? Most mammals have tails. Humans and our ape cousins do not have them. In a new study, scientists have identified a unique DNA mutation shared by humans and great apes, which they say may have contributed to the loss of tails in their ancestors 25 million years ago. Genetic changes in our ancient ancestors may partly explain why humans do not have tails like monkeys. Scientists from New York University Grossman School of Medicine have identified the DNA mutation that is responsible for this. This is a genetic change from about 25 million years ago caused by transposons, also called wandering genes, or jumping genes, which moved to a different position in the genome and changed the way monkeys produce a protein crucial for the development of their tails. Our research explains how evolution removed our tails. This question has intrigued me since my early years, says Bo Chia, the lead author of the study published in the journal Nature. Tens of millions of years ago, the common ancestors of primates, including humans, had tails. Many modern primates still have them, but as they diversified and evolved, some of our ancestors lost their tails. What people were left with was their tailbone. Why do some primates keep their tails and humans don't? The loss of the tail occurred in our ancestors at about the same time that they developed a more upright gait and a tendency to use only 2 of the 4 limbs to support the body. However, finding the genetic basis for this trait was not easy. In 1927, Ukrainian researcher Nadine Dobrovolskaya-Zavodskaya described a strain of short-tailed laboratory mice that she proposed carried a mutation in a gene called T, whose human equivalent is now known as TBXT. Shea began studying this gene known for its role in tail development. A genome search showed that humans and non-tailed primates have a change in the TBXT gene, a short DNA sequence not seen in monkeys with tails. In a next step, the research team used CRISPR gene editing technology to replicate this mutation in mice to see if insertion, a spontaneous mutation in which short DNA sequences are inserted within a single gene, could lead to changes in the protein that controls tail development. The genetically modified animals had a number of tail defects. In some, the tail was shortened or completely missing. In other cases, it was strangely bent or unusually long. But in the course of research, it turned out that there are well over 100 genes related to tail development in various vertebrate species. The authors of the study hypothesized that the loss of tails occurred as a result of mutations, changes in the DNA code of one or more of the genes. They analyzed 140 genes involved in tail development and identified thousands of genetic changes unique to great apes that may also have played a role in tail loss. They identified an intriguing genetic mechanism. Many genes combine to allow the tail to develop. Researchers have determined that tailless primates have an extra transposon, sometimes called a hopping or wandering gene. It is a DNA sequence that can change places within the genome of the same cell, a process called transposition. It is possible that these are remnants of ancient viruses that have become embedded in our genome. They are common in humans and constitute about 10%. Our DNA. Transposition causes mutations and can change how genes function. According to the study authors, it was the insertion of a transposon into the TBXT gene millions of years ago that created the mutation that resulted in the loss of our tails. In the course of evolution, there was a division in the group of great apes, which also included modern humans. Fewer caudal vertebrae developed, giving rise to the coccyx. Some researchers suggest that this change may have favored life on the ground rather than in the trees. But genes often influence more than one body function. So changes that are beneficial in one place may be harmful in another. During the study, scientists noticed a slight increase in the incidence of neural tube defects in genetically modified mice that had the TBXT gene implemented. The neural tube is the embryonic tissue from which the spinal cord and brain develop. Observed defects included spina bifida and an encephaly, no brain.